नमस्कार दोस्तों, बी बॉस डीवल चैनल पे आपका स्वागत करते हैं। आज का हमारा टॉपिक है जो कि बॉटनी से रिलेटेड है, जिसका नाम है काइनेटिक्स इन ग्रोथ। तो चलिए दोस्तों शुरू करते हैं। इस टॉपिक की शुरुआत हम सीड जर्मिनेशन से करते हैं। फर्स्ट ऑफ ऑल, द डेफिनेशन ऑफ सीड जर्मिनेशन. Germination is a process by which the embryo in the seed becomes activated and begins to grow into a new seedling. The types of seed germination. Type 1, the Epigeal Germination. In seeds with epigeal germination, the cotyledons are brought above to the ground to the soil due to elongation of the hypocotyl. In castor, sunflower, cotton, papaya, onion, flat green leaf-like cotyledons can be seen in the young seedlings. Here, the cotyledons, besides food storage, also performs photosynthesis till the seedlings become independent. In some other plants like bean, the cotyledons being thick do not become leaf-like. They shrivel and fall off after their food reserves are consumed by the seedling. Second is hypogeal germination. In this kind of germination, the cotyledons do not come out of the soil surface. In such seeds, the epicotyl, that is the part of embryonic axis between plumule and cotyledons, elongates, pushing the plumule out of the soil. All monocotyledons show hypogeal germination. Among dicotyledons, gram, pea, groundnut are some common examples of hypogeal germination. This is a diagram which showing distinction between the epigeal and hypogeal type of germination. You can pause the video and see the diagram. Now, the factors affecting seed germination. First, the external factors. In external factors, now first is water. A dormant seed is generally dehydrated and contains hardly 6 to 15% water in its living cells. The active cells, however, require about 75 to 95% of the water for carrying out their metabolism. Therefore, the dormant seeds must absorb external water to become active and show germination. Besides providing the necessary hydration for the vital activities of protoplasm, some water softens the seed coat, causes their rupturing, increase permeability of seeds, and converts the insoluble food into soluble form of its translocation to the embryo. Water also brings in the dissolved oxygen for the use by the growing embryo. Second factor is oxygen. Oxygen is necessary for respiration, which releases the energy needed for growth. Germinating seeds respire very actively and need sufficient oxygen. Initially, the germination process is anaerobic due to non-availability of oxygen. Electronic microscopy reveals that mitochondrial cristae are scanty in dormant seeds, which are formed in early stages of germination. The germinating seeds obtain this oxygen from the air contained in the soil. It is for this reason that most seeds sown deeper in the soil or in the waterlogged soils, that is oxygen deficient, often fail to germinate due to insufficient oxygen. Plowing and hoeing irrigate the soil and facilitate good germination. Third factor is suitable temperature. Moderate warmth is necessary for the vital activities of the protoplasm and therefore for seed germination. Though germination can take place over a wide range of temperature, that is 5 degrees Celsius to 40 degrees Celsius. The optimum for most of the crop plants is around 25 to 30 degrees Celsius. The germination in most cases stops at 0 degrees Celsius and 45 degrees Celsius. Light. Some light sensitive seeds require light as an important factor. The seeds which require light are positively photoblastic seeds, for example, lettuce seed. Some are not influenced by the presence or absence of light, that is, non-photoblastic seeds, for example, maize, bean. But some seeds are inhibited by the light, that is, negatively photoblastic seeds, for example, onion, lily. Second type of factor is internal factors. It includes the seed dormancy due to internal conditions and its release. In some plants, the embryo is not fully mature at the time of seed shedding. Such seeds do not germinate till the embryo attains maturity. The freshly shed seeds in certain plants may not have sufficient amount of growth hormones required for the growth of embryo. These seeds require some intervals of time during which the hormones get synthesized. The seeds of almost all the plants remains viable or living for a specific period of time. This bioavailability period ranges from a few weeks to many years. Seeds of lotus have maximum bioavailability period of 1000 years. Seeds germinate before the ending of their viability periods. In many plants, the freshly shed seeds become dormant due to various reasons like the presence of hard, tough and impermeable seed coats, presence of growth inhibitors and the deficiency of sufficient amount of food, minerals and enzymes etc. Next topic is the seed dormancy. Many kinds of seeds apparently ripe fail to germinate even if placed under such conditions that all the environmental factors are favorable. In such seeds, resumption of growth by the embryo is arrested by the conditions within the seed themselves. The state of inhibited growth of seeds or the plant's organs as a result of internal causes is usually called dormancy, but is sometimes referred to as a rest period. Seed dormancy is of considerable advantage to the plant. It enables the embryo to safely pass the unfavorable part of the year and germinate when the conditions are suitable for the establishment of seedlings. In nature, dormancy period coincides with the unfavorable period of the seedlings of the species. So this is the brief of seed dormancy. That factors, first is environmental factors and includes negative or positive light requirement, high temperature, absence of water. Second factor is an internal factor. It includes seed coat with no gas exchange and seed coat with mechanical effects, immature embryo, low ethylene, inhibitors present, that is coumarin, peroxycorbic acid, etc. And next is growth promoters absent. Third type of factor is stimming mechanism. After ripening, loss of inhibitor, appearance of growth promoter. So, a dormant seed is one that is unable to germinate in the specified period of time under a combination of environmental factors that are normally suitable for the germination of the non-dormant seed. Dormancy is a mechanism to prevent germination during unsuitable ecological conditions when the probability of seedling survival is low. True dormancy or innate dormancy is caused by the conditions within the seed that prevent germination under normal ideal conditions. Often, seed dormancy is divided into two major categories based on what part of the seed produces dormancy, that is exogenous or maybe endogenous. There are three types of dormancy based on their mode of action, that is physical, physiological and morphological. So now we are concerning with the factors affecting seed dormancy. First, impermeability of seed coats to water. The seed coats of many species are completely impermeable to water at the time of their maturity. This condition is very common in the seeds of many legumes, of the water lotus and of the morning glory. Germination fails to occur until water penetrates throughout the seed coat. Second factor is mechanically resistant seed coats. In some seeds, as those of mustard, pigweed, shepherd purse, etc., the seed coats are so strong that they do not yield to the pressure of the expanding embryo. The embryos of these seeds have no dormant period and will grow readily if the seed coats are removed. Third factor is the seed coat impermeable to oxygen. The two seeds in the fruit of cockplour are not equally dormant. Under natural conditions, the lower seed usually germinates in the spring following maturity, while the upper seed remains dormant until the next year. Dormancy of these seeds has been demonstrated to result from the impermeability of the seed coats to oxygen. Factor fourth is the rudimentary embryos. In plants like ginkgo, European ash, holly, and many orchids. The embryo is unorganized when the seed is shed and attains full development before it germinates. Fifth factor is dormant embryos. In many species, although the embryos are completely developed when the seed is ripe, the seed fails to germinate even when environmental conditions are favorable. Dormancy of such seeds is a result of the physiological condition of the embryo. Embryo of such seeds will not grow when the seed first ripen, even if the seed coats are removed. During the period of dormancy, some physiological changes called after ripening occur in the embryo before the seed is capable of germination. The seeds of apple, peach, iris, and pine belong to this group. In nature, after ripening occurs in winter and the seeds formed in autumn germinate the coming spring. Factor 6 is germination inhibitors. Germination of seed is sometimes checked or prevented by the presence of compounds known as inhibitors. These are often substances produced in one or more plant organs. For example, the juice of tomato fruits inhibits the germination of tomato seeds, and many other seeds as well. Inhibitors may be present in the embryo, in the endosperm, or in the seed coat. Abscisic acid is one of the most commonly defected inhibitors of the germination. Methods to break seed dormancy. First is mechanical scarification. Seeds having very hard seed coat when scattered with some abrasive material softens the coat and makes it previa to water. Second is chemical stratification. Dipping of seeds in strong acids like sulfuric acid or some organic solvents like example acetone, alcohol, even dipping in boiling water breaks the dormancy by weakening the seed coat. In some instances, seeds subjected to high oxygen pressure also germinate. Fourth, immature embryo dormancy may be broken when the embryos are allowed to complete their development. Fifth, through the application of some chemical like potassium nitrate, gibberellin, kinetin, etc., it is possible to counteract the inhibitors on the seed germination. Cyanide, azide, thiourea also induce germination. On soaking the seed, P-glucosidase become active and cleavage glucose molecules from amygdalin. The glycone releases is decomposed into hydrogen cyanide and benzaldehyde. Significance of seed dormancy. Dormancy in plants both useful and harmful to man. Cereal grains having temporary dormancy which is helpful for the harvesting, dry storage and as a source of food. In the absence of dormancy, these grains were likely to germinate in the field and experience unfavorable conditions. Seeds of several weeds remain dormant for many years and a source of nuisance to the farmers. The survival of species, dormancy has a great advantage, especially in the areas having well-marked seasonal changes and where the winter and summer are of drastic types. In several the alpine and temperate species, budding seed dormancy is initiated with the beginning of winter, and this process helps the plant species to tide over adverse winter conditions without any damage. Similarly, in desert, most annual species have their seed dormant till sufficient rainfall occurs. The availability of rainwater helps in the diffusion of inhibitors present in the seed coat of such species and germination is initiated. So, आज की वीडियो हम यहाँ पे ख़त्म करते हैं और नेक्स्ट वीडियो में हम आपको बताएंगे ग्रोथ के बारे में। वीडियो अच्छी लगी हो तो दोस्तों शेयर करना, सब्सक्राइब करना हमारे चैनल को। नमस्कार दोस्तों, बी बॉस डिवल चैनल पे आपका स्वागत करते हैं।